So in these sessions we'll be solving certain assignment questions, and it will help you to solve the assignments that are given every week with the lecture series. Okay, so feel free to ask any doubt you have regarding the questions, and you can either unmute your chat and then ask me the questions, or you can write in the chat box, whatever is comfortable for you. Just let me admit everyone who are joining the meeting, and then we'll start. So today's session is all about so you might have gone through the video series that have been uploaded on the NPTEL portal. So the lectures are about good lab practices, and then you have certain idea about what are buffers and how buffer solutions are prepared. So we'll solve questions related to those topics. Okay, so we'll see how what kind of questions can be framed on that topic, and I think you have to submit the assignments also, so this session will help you to solve those questions. You can at any point when we are solving the questions, if you have any doubt, you can stop me and ask. Don't be shy. I'll try to help you as much as possible. Okay. Okay, so let's start. So I'll be solving certain questions, and then based on those questions, like these questions are related to your current assignments only, so it would be help you to relate with those. Okay, so starting with the first question, what is the concentration of H plus ions in pure water? So if anybody knows how to solve this question, or anybody wants to answer the question, you can answer it in the chat box. Okay, we have an answer. Aditya says C. Okay, Manushri is asking, how are we accessible to previous recorded problem solving sessions? So after these sessions are over, I am recording the whole session. So after these sessions are over, I'll be uploading the link of these sessions. YouTube, I'll upload the recording on YouTube, and I'll share the link on an Excel sheet. That Excel sheet will be shared on the NPTEL portal, the portal where you are seeing all the questions and all the videos. There only you will I think it is not yet uploaded. So once it is uploaded, you will see a section called problem solving session or live session. So under that section you will have the Excel sheet where all the TAs who are taking these problem solving sessions, so apart from me there are 2 other TAs I guess who are taking these sessions. So all of them will be uploading their recordings, and you can access the videos anytime you want. Okay, those videos will always be available on YouTube. Yeah, so coming back to the question, what is the concentration of H plus ions in pure water? We have an answer from Aditya. It's option C. Okay, that is the correct answer, but let's see how do we land on that answer. So for water, for pure water, we know the molecular weight of water is, what is the molecular weight of water? Molecular weight of water is 18 grams per mole, right? We have water molecule as H2O. So H2O, 2 hydrogen and 1 oxygen atom, the molecular mass is 18, and water dissociates into H plus ions and OH minus ions. Now the concentration of pure water is given as 55.5 millimolar, 55.5 molar. That is basically you divide the amount of water by its you divide the amount of water by 1 liter of volume. So you have 1000 upon 18 or 1 upon 0.018. So that gives you 55.5 molar. So this is the concentration of water. Now what 1 mole of water is giving you 1 mole of H plus ions. So that means concentration of H2O will be equal to concentration of H plus ions. Now at any point we have the equilibrium constant K equilibrium for water is given as 1.8 into 10 raised to power minus 6. So at any point if I have to derive the equilibrium constant of this reaction, that is dissociation of H2O into H plus and OH minus ions, I can define this equilibrium constant as H plus ion concentration multiplied by OH minus ion concentration divided by the concentration of H2O. Now there is another factor that is defined as Kw. How do we define Kw? Kw is defined as H plus ion concentration multiplied by OH minus ion concentration, and that value of Kw is given as 10 raised to the power of minus 14. We can derive this value of 10 raised to the power minus 14 by replacing the value of K equilibrium and the concentration of H2O. And this 10 raised to the power minus 14, now at any point the concentration of H plus ions and concentration of OH minus ions will be equal. Now since concentration of H plus and OH minus ions are equal and their product is equal to 10 raised to the power minus 14, that means concentration of H plus ion is 10 raised to the power minus 7, which is equal to concentration of OH minus ion. So at any point, if anybody asks what is the concentration of H plus ions in the pure water, it is 10 raised to the power minus 7 molar, and that is why the pH of pure water is equal to 7. Because how do we define pH? We define pH as minus log H plus ion concentration, right? Minus log of H plus ion concentration. Now if my H plus ion concentration is 10 raised to the power minus 7, that means pH will be minus log of 10 raised to the power of minus 7. If you will solve this, you will get the answer 7. So that is why pH of pure water is 7. Okay. I hope this is clear. So moving to the next, if anybody has any doubt, you can ask me. Okay, if there is no doubt, then we will move to the next question. Question number 2. So question number 2 is, while working with radioactive isotopes, radioactive shields made of dash are used for protections. Option A, lead, option B, potassium, option C, boron, and option D, platinum. So what is the material which is used for making the radioactive shields? Now think about a material that can be used for making radioactive shields, that means that can absorb the radiations, that can protect you against the radiations. Okay, so we have the options lead, potassium, boron, and platinum. Anybody knows the answer? Okay, we have few answers here. Okay, everyone is saying option A, lead. So yes, that is the correct answer, lead. So why do we use lead for making these radioactive shields? What is so special about lead that is not there in potassium, boron, or platinum? Yeah, I think Shaheen wants to answer something. Let's see. Yes, exactly. The correct answer is high density. Because of its high density, it can protect us against the radiations. So basically, lead is a highly dense material, and because of that high density and high atomic number, it has high number of electrons. And once it has high number of electrons, it makes it ideal for shielding in most of the medical radiation environments. So, since it has the high density, it is very effective at reducing the radiation exposure. So, it can basically, it can effectively reduce the radiations, and thus used in making all the shields that can protect you against the radiation. Okay, so in a medical setup where someone is working with x-ray machines, or in a lab setup where you have to work in a radiation prone environment, so in that case you have to wear proper protective equipment, protective kits, and such equipments are made to protect you against a harmful environment. Okay, so something that can actually affect you, something that can actually affect your health. So to prevent that, just a second, just give me a second. Is my presentation visible?
Yes, madam.
Yeah, just give me a second. I think now it is visible, right? Yeah, sorry for that connection issue. Just give me a second. I'll okay. Let's start. Sorry for that. Uh, so that is why we use certain kinds of protective equipments when we are working in a hazardous environment that can affect our health. To protect ourselves, we wear such equipments. One of the equipment is a radiation shield, if in case you are working in a radiation prone environment. Okay. Okay, moving on to the next question. Biosafety level 3 is used for organisms that are dash. So option A is cannot spread through aerosol in lab setting. Option B is are exotic and pose high risk of fatal aerosol transmission. Option C is organisms that are potentially lethal and can spread through respiratory transmissions, and option D is organisms that are not known to be lethal to laboratory personnel and environment. So for which category we will use BSL-3? Okay, we have the answer option C, organisms that are potentially lethal and can spread through respiratory transmissions. Yes, that is the correct answer. We will use BSL-3 for such organism. So we have actually 4 classes of biosafety cabinets or biosafety. We have 4 levels of biosafety. So we have BSL-1, BSL-2, BSL-3 and BSL-4. So based on the increasing risk factor, we increase the number in this biosafety level. So for BSL-1, since it's the lowest risk, since it is at the lowest risk, it is categorized as the first biosafety level, and mostly it is used for all the organisms that have no disease causing that cannot cause any disease, very low risk agents. So these organisms are generally not known to cause human diseases. Okay, so best example is E. coli. It's a most commonly used bacteria that is used in all the biotech labs. So any biotechnology or any molecular biology work if you want to do, this is the best organism. It grows very quickly, it is not harmful to humans, they do not cause any diseases, and it is very easy to use. So that is why it is at the lowest risk, and we can use a biosafety level 1 cabinet for handling such organisms. Okay, then we move to the next risk factor, the next safety level, that is BSL-2. So when the risk is slightly increased, now what do we handle in BSL-2? So in BSL-2 category, basically all the infectious agents of moderate risk. So it's not high risk, but it's not low risk. So it's somewhere in between. So infectious agents of moderate risk with ingestion or mucous membrane transmission. Okay, so it can cause risk or it can cause infection either if it is ingested or it is actually transferred through a mucous membrane. Okay, so all the influenza and Lyme diseases and salmonella, measles, these, so some things that cannot pass through aerosol. Okay, I hope you understand what aerosol means. So just by coughing, and like if I am infected and if I cough and the cough droplets are transfers through the air to another human being, so that means it is transmitting through aerosol. Okay, so in such cases we have to go to a higher biosafety level. So things that are not transmitted through aerosol, things that are transmitted through ingestion or through mucous membrane transmission, that can be handled in BSL-2. So from BSL-2 onwards we are handling infectious agents, but BSL-2 includes things that cannot be transferred through aerosol. BSL-3 includes the infectious agents that can be transferred through aerosol transmission. That means it can transfer through air, and the best example that we all have gone through is coronavirus. Now you know that it can spread through aerosol. So coronavirus, and then Mycobacterium tuberculosis, that is the causative agent of TB. We have Yersinia pestis, and then malaria. All these infectious agents, they can be transmitted through aerosol, and thus if you are working on these in a laboratory, you have to have a BSL-3 equipped setup. Okay, so you have to have a lab that is equipped with all the safety requirements that comes under biosafety level 3. Okay, similarly the highest risk factor, that is BSL-4. So now we have already covered all the infectious agents that are transmitted through aerosol and that are transmitted through ingestion or mucous membrane. So what is left now? So what is left is there are certain infectious agents whose cure is not available yet, or whose risk factor is not known yet. So there are infectious agents that can transmit through aerosol, and we do not know the exact treatment of that disease. Like, in case I get that infection, I don't know how to treat myself. So for those particular agents, we have to have a BSL 4 level. BSL 3 won't suffice, we need a BSL 4 level. So Ebola virus, variola virus, we don't have any treatment available, and these are lethal infection, so they can ultimately lead to death. Okay, so for these lethal infections, we have to have a BSL 4, that is the highest biosafety level. Okay, so I hope you understand all the biosafety levels. So we have BSL 1, 2, 3 and 4, and based on the increasing risk factor, we are based on the increasing risk factor, basically the biosafety level increases. Okay, what is Marburg virus? It is a kind of virus. It causes a disease, like it also causes something similar to like it causes a disease that can be transmitted through aerosol transmission only. I don't exactly know the name of the disease, but all these viruses, they don't have any treatment available, and they are basically lethal viruses. Like, the infection caused by them are lethal. They can ultimately lead to death since there is no treatment available. Okay, I don't exactly know the disease caused by Marburg virus. Okay, okay, so I hope all the biosafety levels are clear. Any doubt in any doubt in this? Okay, moving on to the next question. Question number 4. We have to tell the amount of NaOH that is required to make 350 ml of 4 molar NaOH solution. Molecular weight of NaOH is already given. So you have to calculate this. I can give you time to calculate this, and meanwhile we all can calculate together. So we have to make 350 ml of 4 molar NaOH solution. Okay, molecular weight of NaOH is given as 40 grams per mole. What do you mean by this? Molecular weight of NaOH is 40 grams per mole. That means 1 molar NaOH solution will have 40 grams of NaOH in 1 liter, of water. Okay, if we are making a NaOH solution, we will make it in water. Okay, so 1 molar NaOH solution will have 40 grams of NaOH in 1 liter of water. Why? Because the molecular weight of NaOH is 40 grams per mole. That means 1 mole is equivalent to 40 grams of NaOH. And how do we define molarity? Molarity is defined as moles per liter. Okay, moles per liter. So if I have 1 molar NaOH, that means we have 1 mole of NaOH in 1 liter. 1 mole of NaOH is equivalent to 40 grams of NaOH in 1 liter. This is clear? So now how much we have to make? We have to make 4 molar NaOH. Now if 1 molar NaOH has 40 grams in 1 liter, that means 4 molar NaOH will have 4 into 40 grams, right? If 1 molar has 40 grams, 2 molar will have 2 into 40 grams. Like that, 4 molar will have 4 into 40 grams in 1 liter of water. Okay, do not forget this. 1 liter of water. Now the next part is how much volume we have to make. We have to make 350 ml. So now we will see in 1 liter solution we are having 4 into 40, that means 160 grams of NaOH, right? That means 1 liter is equivalent to 1000 ml, right? 1 liter is equivalent to 1000 ml. So 1000 ml water has 160 grams of NaOH. That means 350 ml will have 350 ml water will have, or 350 ml solution will have 160 divided by 1000 into 350, right? 160 grams divided by 1000 ml into 350 ml. So solve this and we will get our answer. Sorry. Yeah, solve this and we will get our answer. So 160 divided by 1000 into 350 will be equivalent to 56 grams. Okay. So the correct answer is option A. Okay. So 56 grams of NaOH will be required to make 350 ml of 4 molar NaOH solution. Okay. Shaheen had answered 56. Very good. So you reached the correct answer. So correct answer is option A. Any doubt in this question? So these are the kind of problems that you will have to solve if you are making any kind of buffer or any kind of solution in a biotech lab. Okay. So whenever you are working in any biotechnology experiment or a molecular biology experiment, these solutions are something that you have to routinely prepare. Okay. And if I have to prepare different molarity solutions of NaOH, I should not spend a long time to think how much grams I have to weigh and then how much water I should add to make this particular concentration. So that's why if you practice such questions, if you keep practicing such questions, it would be very easy for you to quickly find out the answer. So this is very simple calculation. Just if you know the molecular weight of any compound, you can easily know that okay, that much gram will go in 1 liter of water to make 1 molar solution. Okay, whatever the molecular weight is, molecular weight corresponds to the weight in 1 mole of that compound. So weight equivalent to 1 mole of a particular compound is the molecular weight. Okay, it's written as grams per mole. That means 1 mole has that particular grams. Okay, once you have the molecular weight, you can easily calculate the molarity for 1 liter. Now if you know the molarity for 1 mole, 1 molar and 1 liter, you have 3 factors. Now whatever is given in the question it was 4 molar, so we calculated for 4 molar. In the question it was 350 ml, so we calculated for 350 ml. Okay, I hope there is no doubt in this. Okay, so let's move to the next question. Question number 5. In case of diabetes and fasting, dash is released which causes acidosis in stomach. Option A, hydrochloric acid. Option B, beta hydroxybutyric acid. Option C, butyric acid. And option D, citric acid. Anyone knows? Okay, Tanisha has answered option B, beta hydroxybutyric acid. Okay, that is the correct answer. So in case of diabetes and fasting, yeah, we have more answers. Everyone is answering option B. So that is the correct answer, beta hydroxybutyric acid. So in case of diabetes and fasting, mostly beta hydroxybutyric acid is released and it causes acidosis. And acidosis is basically decrease in the pH. So it is creating an acidic environment, and because of that acidic environment, it causes a lot of symptoms like headache, nausea. Okay, so yeah, so beta hydroxybutyric acid is the correct answer. Okay. Moving to the next question. In the reaction given below, identify the conjugate acid for the base NH3, that is ammonia, which dissociates in the buffer system as follows. So ammonia reacts with HCl to form ammonium ion and a chloride ion. Okay. So tell me what would be the conjugate acid for the base ammonia. Can you once again move to the previous slide? Yeah, Manushri, I can show you the previous slide. Do you have any doubt, Manushri? Okay, we have the answer for the next question, option C. Okay, I'm moving to the next question now. So in this question, we have to tell the conjugate acid for the base ammonia. So everyone is answering option C, that is ammonium ion. Yes, that is the correct answer. So here we have HCl as the acid and ammonia as the base. So whenever an acid and base reacts, we get a conjugate acid and a conjugate base associated with that. So here the HCl is releasing the chloride ion and ammonia is releasing the ammonium ion. Basically, HCl is losing the proton to release the chloride ion, and ammonia is accepting that proton. This proton is given to ammonia. So this proton is accepted by ammonia, ammonia being the base. So you must have studied acids are proton donor and bases are proton acceptor, right? Right? So ammonia being the base will accept the proton from the acid HCl and get converted into ammonium ion. Now if we have ammonia as the base, its conjugate acid will be ammonium ion. If we have HCl as the acid, its conjugate base will be chloride ion. Okay, so chloride ion will be the conjugate base and the ammonium ion will be the conjugate acid. So conjugate acid for the base ammonia is ammonium ion, that is option C. Okay, most of you answered it correctly. Anyone has any doubt in this? So you understand what is a conjugate acid and a conjugate base? Any base, if once it accepts the proton, the product that is forming will be its conjugate acid. Okay, why it is called the conjugate acid? Anyone knows how it is acidic? Can anyone answer how it is, why it is called conjugate acid? Any idea? No? So if you see here, ammonium ion now has an extra proton, right? NH4 plus. So it has an extra proton. So now it can act as a proton donor. I told you acids are proton donor and bases are proton acceptor. So now ammonium ion, if you see the reverse reaction, see it is a reaction that is happening in equilibrium. So this is the forward reaction, and if you see the reverse reaction, that is ammonium going back to ammonia. In that case, this NH4 plus will donate its proton to the chloride ion, right? So this, in that case, ammonium ion will become the proton donor and chloride ion will become the proton acceptor. That's why the ammonium ion is the acid here and chloride ion is the base. Okay, so conjugate acid and conjugate base. Okay, I hope now it is clear. Okay, let's move to the next question, question number 7. During titration of a weak acid with strong base, which of the following will be observed? So during titration of a weak acid with a strong base, which of the following will be observed? Option A, pH will keep on increasing constantly. Option B, pH will increase rapidly initially and then slowly near pK value of acid. Option C, pH will not increase, and option D, pH will increase slowly initially and sharply near pK value of acid. So what would be the correct answer? Yeah, Tanisha says B. Okay, we have more answers here. B. pH will increase rapidly initially and then slowly near the pK value. Yes, that is the correct answer. So what do you mean by pK? Can you answer? What is pK value? What does pK stands for, or what does it denote? Dissociation constant. Yes, very good. So dissociation constant of an acid is given by pKa value. So since it's a weak acid, what happens is when you keep on adding base, so if we have a weak acid HA, okay, so it will dissociate into H plus and A minus. Okay, so if I have a weak acid and if I keep adding strong base, initially what will happen? Initially the pH will rapidly increase. Why? Because since it's a strong base, the dissociation won't start quickly. Since it's a weak acid, actually dissociation won't start very quickly. Just a second. Yeah, since it's a weak acid, dissociation won't start very quickly. So initially when I will add the base, the OH minus concentration will increase, and if OH minus concentration will exceed the H plus ion concentration, the pH will shift towards the basic side. So whatever the pH of the solution is, if I add a strong acid, the OH minus concentration will suddenly increase and the pH will shoot up. Okay, but what happens when it reaches near the pK value, where the dissociation constant, where the acid is actually going to dissociate? Okay, so let's say I have a curve here. Okay, if I have a curve, so the curve will go something like this. So once the pH, so this will be the pH scale. Okay, so as I keep on adding OH minus ions, the pH will increase rapidly. Okay, I'm adding the base, I'm increasing the pH, so pH will sharply increasing. But when it reaches its pK value, at the pK value, since it is the dissociation constant, this dissociation will start happening. Okay, when this dissociation is happening, what will happen? H plus ions are released. Now if H plus ions are released, they can react with the OH minus ions to form H2O, right? H plus ions can react with the OH minus ions to form H2O. So basically H plus ions can neutralize those extra OH minus ions that are being added from the strong base. Okay, so a weak acid, when we are adding a strong base to it, initially the pH is increasing because of the OH minus ions. Initially the pH increases rapidly because of the OH minus ions that are released from the strong base, but slowly near the pK value of acid, because near the pK value of acid, the acid is dissociating. The acid is dissociating and releasing H plus ions. These H plus ions are now neutralizing the OH minus ions. Since it is neutralizing the OH minus ions, the pH will now slowly increase, or it will kind of get constant in that particular region. Again, once you exceed the pK value and keep on adding base, again the pH will shoot up. After a certain point, if you don't stop adding the base, again the pH will shoot up. Okay, so I hope this is clear. I hope this is clear why the pH is increasing rapidly initially and then slowly near the pK value. What do you mean by pK value, and what is happening in case of a weak acid? Okay, when we say weak acid, basically we mean that it is going to dissociate slowly. If we have a strong acid, that's why they have mentioned it's a strong base. When we are adding a strong base, it is quickly dissociating. As soon as we are adding the base in a solution, it is quickly dissociating. So an example of a strong base is NaOH. If I add NaOH to a solution, it will quickly dissociate into Na plus and OH minus. Okay, but a weak acid like let's say acetic acid, so acetic acid won't dissociate quickly into acetate ions and proton. Okay, so for that to happen, protons will be released slowly from the acetate ion, and that is why they are called weak acids, because they dissociate slowly. Their pKa is at the higher side. Okay, I hope that is clear. Let's move on to the next question now. For the given chemical reaction, HNO3 plus H2O gives H3O plus NO3 minus. Henderson-Hasselbalch equation is expressed as follows. So we have 4 options here. pH is equal to pK plus log NO3 concentration divided by HNO3 concentration. Option B is pH is equal to pK plus log HNO3 concentration divided by NO3 minus concentration. Option C says pK is equal to pH plus log NO3 minus concentration divided by HNO3 concentration, and then option D says pK is equal to pH plus log HNO3 concentration divided by NO3 minus concentration. So for this given chemical reaction, what would be the Henderson-Hasselbalch equation? Let's see. Yeah, so we have answers from 4 people. Everyone says A. Option A, pH is equal to pKa plus log NO3 minus divided by HNO3. Yes, that is the correct answer. So we have here HNO3, that is, what is HNO3? It is nitric acid, right? So nitric acid in the solution is dissociated into a hydronium ion and a nitrate ion, right? So now, like I told you about acid and its conjugate base, right? So we have here HNO3, that is nitric acid, as the acid. So what would be the conjugate base? Conjugate base will be the nitrate ion, right? So conjugate base, or we can say salt, okay, conjugate salt. So any Henderson-Hasselbalch equation is actually denoted as pH equal to pKa plus log of salt concentration divided by acid concentration. Okay, so log of salt concentration upon acid concentration. So that is why option A is the correct answer. We have log of NO3 minus, that is the conjugate salt, divided by the acid, that is HNO3. Okay, log of salt concentration divided by concentration of acid. Okay, this is clear. Okay. So correct answer is option A, pH is equal to pKa plus log NO3 minus concentration divided by HNO3 concentration. Moving on to the next question, question number 9. pKa can be defined as dash. Option A, pH at which 100% acid is 100% acid is ionized. Option B, pH at which 50% acid is ionized. Option C, pH at which 50% base is ionized, and option D, pH at which 100% base is ionized. So I had already asked you what pKa is, and you told me it's the dissociation constant. So, and I already explained in the previous question what dissociation of acid means. So you should be able to answer the question. I see only 3 or 4 people are answering continuously. I want answers from everybody. This is a live interaction session. It would be very helpful if you also interact along with me. It would be helpful for me. Okay. So we have the answer B, option B. pH at which 50% acid is ionized. Okay. So pKa can be defined as the pH at which 50% acid is ionized. That is the correct answer. So I told you pKa means the dissociation constant. So any dissociation constant for a acid. Similarly, we have another term called pKb. Okay, that is the dissociation constant for a base. So for pKb, it would be pH at which the 50% base is ionized. Okay, so pK is the dissociation constant of acid at around the the pK value is basically the pH at which the acid starts dissociating. Okay, and the pK would be basically the midpoint of the slope that I showed you. So the midpoint of the curve where the acid starts dissociating. Okay, so if I have a acid called HA and it is dissociating into H plus and A minus, that is, it is releasing its proton. So at any point, if I keep on adding the base to this acid, it will keep on dissociating and the pH will increase, but there will be a pH where only 50% of acid will be in this form and 50% of acid will be in this form. Basically, the reaction will be in equilibrium. Okay. So in that case, we will at that particular pH is defined as the pKa for that particular acid. So every acid has its own pKa value. If we have a acid that can have we can have acids that can have 3 different pKa values also. Example is phosphoric acid. Since it has 3 protons, it can release 1 proton at a particular time. Okay, so phosphoric acid can actually release 1 proton and become H2PO4 2 minus, and then it is H3PO4 not minus. So H3PO4 can become H2PO4 minus, and then HPO4 2 minus, and then PO4 3 minus. Okay, so in that case, every time an acid is releasing a proton, it will have a particular pKa value for that particular proton. So every proton will have its own pKa value. Like at that particular pH, that particular proton will be ionized. Okay, I hope this is clear. the concept of pKa. Okay, let's see the next question now. Question number 10. For preparing a buffer, just a second. Yeah. For preparing a buffer of pH 6.5, what is the ideal pKa range of the acid to be used? Okay. So for preparing a buffer of pH 6.5, what is the ideal pKa range of the acid to be used? Option A 7.5 to 8.5, option B 4.5 to 6.3, option C 2.5 to 9.5, and option D 5.5 to 7.5. So what would be the correct answer? Option D 5.5 to 7.5. Okay, we have option D as the common pole. So yes, that is the correct answer. But why option D is the correct answer? So I have explained about pK in the previous two questions. So I think now you understand what role pK is playing. So basically, what do you mean by a buffer? A buffer is a solution where if you add, yes, Sunita, pH is equal to plus minus of pK. Yes. So a buffer is basically a solution in which if you add a small amount of acid or a small amount of base, the pH will not shift drastically. Okay. So in that case, we need a solution where the pH remains constant in case we add a small amount of acid or a small amount of base. And that will happen, you saw in the previous question we had a solution of weak acid where we were adding a strong base. So what was happening, around the pKa value the pH was slowly increasing. Why? Because at the pKa value the acid starts dissociating and the released H plus ions are strong enough to neutralize the OH minus ions, right? So for any buffer system, so a buffer system is made by a weak acid and its conjugate salt. Okay, so we have Shaheen has written example includes acetic acid. Yes, that is a very good example, so I'll explain with that example only. So acetic acid is basically CH3COOH. Okay, so if I take acetic acid and its conjugate salt, that is sodium acetate, let's say. Okay, if I mix these two, basically I make a solution of sodium acetate and then adjust the pH with acetic acid, I will have a buffer system of acetate ion. Okay, I will have a buffer system of acetate ion. I can call that buffer acetate buffer. What will happen in this pKa range, the pKa range that is 5.5 to 7.5, I can make a buffer with these acetate ions only in that range. Other than that, the pH will not be constant for this particular system. Why? Because at the pKa value, the acid will be 50% dissociated. I told you that 50% of protons will be ionized at the pKa value. Okay, so pK is the pH where we will have the half ionization. We will have 50% proton ionization, right? So 50% dissociation will occur at that particular pH. That means we will have a constant system where the amount of protons released are enough to neutralize any OH minus ions that are added, and also the amount of acetate ions are enough to neutralize any protons that are added. So we have a buffer system in which a small amount of acid or a small amount of base when added to that buffer system will not change the pH. Okay, if I add small amount of acid, that is small amount of H plus ion concentration, I will have enough anions to neutralize this protons. And if I add a small amount of let's say base, what will happen? We will have enough protons to neutralize this hydroxide ion. Okay, so that's why the for making a buffer system for at any pH, we will have to see the pK range. So whatever anion will have the pK range around that particular pH, we will use that particular buffer system. Okay, so for this case we can use this system. Okay, I hope this is clear. Let's move to the next question now. While preparing fresh phosphate buffer of pH 7.4, accidentally the pH overshot to 7.8. What should be ideally used to adjust the pH of the phosphate buffer back to 7.4? So I was preparing phosphate buffer of pH 7.4, but accidentally the pH overshot to 7.8. What should I add to adjust the pH back to 7.4? Option A, 1 normal phosphoric acid, option B, 1 normal hydrochloric acid, option C, 1 normal sulfuric acid, and option D, 1 normal nitric acid. What would be the answer? Shaheen says option B, hydrochloric acid. Okay. Okay, all the answers are option B. Why do you think hydrochloric acid should be used? Hydrochloric acid is not a weak acid, if that is your answer. Can anybody answer why people who have answered option B, why do you think it should be option B? So read the question properly. It's a phosphate buffer system, right? Yeah, Rucha answered option A. Why, Rucha? Why do you think it should be option A? Both of you should have some explanation. Suman, Tanisha, you have answered option B. So I've told you, I've just now explained how a buffer system works, right? I gave you an example of acetate buffer. So for making acetate buffer, yeah, so now you know. Yeah, so now I think everyone understood the question. So for making acetate buffer, what acid I was using? I was using acetic acid. So basically whatever buffer system I am using, I have to keep the same ions in the solution. Otherwise it won't be called a buffer. Okay, a buffer will be made from a weak acid and its conjugate salt. So I can use sodium phosphate or a potassium phosphate salt and its conjugate acid. The acid will be phosphoric acid. Okay, so option A will be the correct answer. The ion system should not change in the buffer, otherwise it won't be buffering in that particular range. Okay, So when I have phosphoric acid in, like if I add phosphoric acid, it will dissociate into phosphate ions and protons will be released, right? So that will keep the system in the same environment. If I now use HCl, what will happen, or H2SO4 or HNO3, all three are very strong acid. So I told you, if I am using any strong acid or a strong base, what will happen, it will quickly dissociate. If I add HCl to the solution, it will quickly dissociate into H plus and Cl minus. What will happen, there will be an overshot of H plus ions basically, and your pH will drastically decrease. So right now my pH is 7.8, right? If I add HCl instead of phosphoric acid or H2SO4 or HNO3, the pH will drastically decrease. It can reach to up to 1. Okay, so that is why you have to make a buffer with a weak acid and its conjugate base, so that the dissociation happens slowly. You can never make a buffer with HCl or H2SO4. You can use it to adjust the pH in some buffer systems where chloride ions are present or where sulfate ions are present, but you can never use HCl as the buffering component. HCl cannot do the buffering component because its dissociation constant is very low, so it will quickly dissociate. But we can use weak acids that will slowly dissociate, slowly release the protons, and slowly maintain the pH in a particular bracket. So if I am making a buffer of 7.4, it should be 7.4 even if a small amount of base or a small amount of acid is added. Why this buffering system is very important is, let's say in your body there are multiple enzymes, multiple proteins that are working, right? So those proteins have a particular optimum pH where they can function properly. Now the proteins that are present in your stomach are meant to work at acidic pH. So now if I suddenly give you overdose of antacids that are basic in nature, your proteins that are present in the stomach will not be able to function properly. Similarly, your blood has a neutral pH, so there is a need to maintain pH around your body, like inside any organism there's a need to maintain that pH for proper function, and that pH is maintained by these buffering systems. So in blood the buffering system is by carbonate, the carbon dioxide that we breathe in, that goes and becomes carbonate ion, and then there is a carbonate buffering system. So similarly, whenever we are doing any experiment, if I am doing a biological experiment where I am using a enzyme and I am doing an enzymatic reaction, I want that enzyme to work in that particular pH where it is most active. Okay, and for getting that particular pH, I will have to take a buffer of that particular pH. Why I am taking a buffer? So that even if the substrate or whatever components I am adding to the reaction mixture, those components should not disturb the pH, and my enzyme should be able to function perfectly in that particular pH, in that particular pH in which I have prepared the buffer. Okay, so that is the use of buffer. Why do we need buffers in our daily life? Is basically because we need to maintain a particular pH for every proteins and every enzymes and every function that is happening in our body. Okay, yeah. So I hope this question is clear now. So this is a request to all of you. This is a request to all of you to read the questions properly, because sometimes it is actually like a very simple question, but because we don't read the question very properly, these small silly mistakes, especially in case of multiple choice questions, these things just slip out of our mind, and this can just lead to losing marks. Okay, so just be careful while reading the questions. So we'll move to the next question now. Question number 12. What is the material safety data sheet? Option A is a catalog of standard operating procedures for lab. Option B, a catalog of standard operating procedures for biosafety cabinet. Option C, a catalog of information about the potential hazard of the material provided by the manufacturer. And option D, a catalog of instructions to be followed if any spillage, accident or injury happens. So what would be the correct answer? What is material safety data sheet? We have answer from Sunita. She says option C, a catalog of information about potential hazard of material provided by the manufacturer. Yes, that sounds correct. Yeah, more people are answering now. Option C. So that is the correct answer indeed. So material safety data sheet, now by the name itself you can see it is a sheet, or it is a piece of information that is telling you about the safety of the material. So with any chemicals, if you have noticed, if you have ever worked in a lab, so you must have noticed in any chemical bottle you have the MSDS if you check any chemical details, like let's say just search NaCl bottle like that is used in laboratory. If you go to any company's website, Sigma, Merck, so if you see their website, you will see a tab saying material safety data sheet. So with every chemical we get a material safety data sheet, MSDS. Okay, and that MSDS tells us about the safety instructions that are required, like that should be known before handling that particular chemical. So if I am handling any chemical and I don't know whether it is toxic, whether it is flammable, or whether it is corrosive, so if I don't know any information and I just handle it randomly, it can actually cause potential hazard to me. Okay, so if I don't know that HCl is corrosive and I just pour it like that and something spills over my hand, I can get severe burns, right? So that is why just a second, Shine. So that is why we need a sheet, we need a piece of information that can tell us that okay, this material is toxic, this material should not be ingested, what should we do in case it comes in contact with our eye, what should we do in case we ingest it. Such informations are given in the catalog, in this piece of information basically, which is called MSDS or material safety data sheet. Okay, Shaheen is asking, ma'am, what is catalog for B? Catalog of standard operating procedures for biosafety. So standard operating procedure is a different set of information. So these are called SOPs. So when you work in any lab, mostly in most of the industrial labs, they have set of SOPs that are called standard operating procedures. So starting from how to enter the lab, you will have a SOP for that. How to wear your protective equipment, the personal protective equipment, the PPE kits, you will have a SOP for that. How to use a particular instrument, that will have a separate SOP. So biosafety cabinet is a type of instrument only. So it's a cabinet where, so you remember I told you about different biosafety levels. So based on different biosafety levels, we have different cabinets that are designed. It has a laminar airflow and HEPA filters attached to it, so that you can handle based on the biosafety level. You will have different protection level in the biosafety cabinet and you can handle those organisms. So if you are working with E. coli, you will have a normal laminar flow hood. If you are working with some infectious agents, you will have some higher level of biosafety cabinet. So those are parts, like those are equipments and instruments. So for every equipment and for every instrument in a lab, you will have a operating procedure that will tell you exactly how to use that, like starting from open the lid of the hood, sterilize the hood with 70% IPS, starting from each and every step. Like if it is a SOP for wearing the PPE kit, it will exactly tell you what all to do, like wear your goggles, wear your mask, sanitize your hands first and then wear your mask, wear gloves. So these are the kind of documents you will see in a lab, in most of the laboratory setups. Okay, so standard operating procedures is a type of document only. So for lab it is different, for equipments it is different. So that's there. Material safety data sheet is completely different. It is mostly associated with the materials, any chemicals, any material that you are buying, you will have a, from the manufacturer's side they will give a MSDS. So let's say I have a company, I can manufacture some chemical, or I am synthesizing a compound that I want to sell commercially. So if I am synthesizing a compound, I should know what are the potential hazards, what and how to deal with it. Okay, so that is your MSDS. I hope that's clear. You all can actually, after the class, I would suggest you to go to any chemical website, just the common ones, and just check for any chemical's name. You will actually see MSDS is attached with each chemical, and you can see how it is written. It will have certain details like the chemical, if it is toxic, non-toxic, if it is an irritant, if it is a corrosive, all these details will be there. So if you are curious enough, you should actually go check out a website, or just Google MSDS for any chemical, and you will find some or the other link that will lead you to it. So that is all about MSDS. Any doubt? Any other doubt? Shine, is your doubt clear now? So we will move to the next question. What is the pH of the solution containing 0.00001 molar OH minus ions? Okay. So option A 2, option B 9, option C 4, option D 7. So you have to solve this question. We have to calculate the pH of a solution that has these many OH minus ions. So the concentration of OH minus ion is given. I'll just write it in. Sorry. Yeah. So how do we solve this question? We have the OH minus ion concentration given here. This is 10 raised to the power minus 1, 2, 3, 4, 5. Right? 10 raised to the power minus 5 molar. So OH minus ion concentration is given. Okay, we have answers here. Option B. Option B, that is 9. Okay, let's see. Let's solve it. So we have OH minus ion concentration given as 10 raised to the power minus 5. So we know that pH is equal to minus log of H plus ion concentration. So we don't have the H plus ion concentration, we have the OH minus ion concentration. So like we can define pH, we can also write POH, right? So POH can be written as log of minus log of OH minus ion concentration, that is negative logarithmic of OH minus ion concentration. So we know the OH minus ion concentration, so we can calculate POH. So that would be minus log 10 raised to the power of minus 5, and if you solve this you will get the answer 5. So POH is 5. Now pH is basically equal to pH is basically equal to 14 minus POH, right? 14 minus POH, because if you remember I told you about a value called Kw that was equivalent to 10 raised to power minus 14. So a pH scale goes from 0 to 14, okay, 14 being the most basic, 0 being the most acidic. So the pH scale goes from 0 to 1 to 14, okay. pH scale goes from 1 to 14. So 14 is the ultimate value. So pH will be, so at any point the concentration of H plus ion and OH minus ion will sum up to 14, okay. So pH plus POH will always be 14. So now we have POH value as 5, so 14 minus 5 is 9. So pH is equivalent to 9. So the option B is the correct answer. You were right. So people who haven't answered, I hope it is clear to you. People who have answered, I am assuming you understood the question and solved it. People who are not answering, if you have any kind of doubt, ask me. If you do understand how to solve it, well and good. I have shown how to solve it. If you want me to again tell you how to solve it, I can. Okay. So basically OH minus ion concentration was given. We calculated the POH. POH is calculated as the negative log of OH minus ion concentration, that is negative log of 10 raised to power minus 5, and you get the answer 5. And then you have pH is equal to 14 minus POH, 14 minus 5 is 9. So the correct answer is option B. That's clear? That's clear to everyone, I hope. So we'll move on to the next question. Okay, question number 14. While pipetting a high viscosity liquid, which of the following technique should be followed? Option A, take the liquid rapidly from the pipette. Option B, cut the microtip from top. Option C, use the mouth operated pipette. And option D, use measuring cylinder. What should we do to pipette out a high viscosity liquid? Option B, cut the microtip from top. Yes, that is the correct answer. So any high viscosity liquid, if you take the liquid rapidly from pipette, you will have a lot of air bubbles, because since the viscosity of liquid is high, it won't get sucked up too quickly. So when you are pipetting it out with auto pipetter or manual pipetter also, you have to since the tip of the microtip is very fine, so it is easier to withdraw liquids like water or any other solution that are of low viscosity. But if you are pipetting out liquids like glycerol, that is very viscous, okay. So for that, in that case, you have to cut the microtip from top in order to make the tip wider, so that you can withdraw the liquid easily in one fluid motion. Otherwise, if you don't cut the tip and if you rapidly try to take the liquid, it will form bubbles. Also, if you try to use a mouth operated pipette, you will have to really suck it hard to withdraw the liquid. That is very difficult. And using a measuring cylinder is also not a good option, because if it's a high viscous solution, it will definitely stick to your measuring cylinder, and then again you will have very error prone solution that whatever you are making. Okay, so the best solution is to cut the microtip from top. Any doubt? Okay, correct answer is option B. Okay, moving to the next question, question number 15. The dissociation of phosphoric acid is shown below along with the respective pKa values. So I told you phosphoric acid can actually dissociate in 3 levels. It can dissociate, like every proton, when it is dissociated, we have a particular pKa value. So H3PO4 dissociates into H2PO4, and then H plus ion is released. That is the first pKa. We have the first pKa value, that is 2.12. Then the second step, where H2PO4 minus, again a proton is released and HPO4 2 minus is released. So you have the second pKa value, that is 7.21. Then HPO4 2 minus, from HPO4 2 minus, again the proton is released and you get PO4 3 minus. You have the third pKa value, that is 12.32. Which of the above pKa value makes it a suitable phosphate buffer system in the pH range of 2.9 to 3.1? Options are option A pK1 and pK2, option B pK1, option C pK3, option D pK1 and pK2. Okay, option A and D are similar, sorry for that. Okay, we have answers. Option B pK1. Okay, it is option B. So we have discussed about pK multiple times now, so I hope this is very clear to you. So for making buffer of any pH, you have to see the pK value. So if I want to make the buffer of pK, if I want to make a buffer of pH 2.9 to 3.1, I have to see around which pK value it is matching. So it is matching the first pK value. If I want to make a buffer, let's say of 7.4, I want to make a phosphate buffer of pH 7.4, I will use the second pK value, that is 7.2. So phosphate buffer system is actually very versatile. Since it has 3 pK values, you can actually make a buffer of 3 different pH using the same components, just in different ratios. If I use more acid component, you will have a buffer of a low pH. If I use both the acid and basic component in almost equal amounts, you will have a buffer at a neutral pH. And if I use the basic component at a higher amount, you will have the buffer at a higher pH. So it can actually give you a buffer system at 3 different range of pH. So it's actually a very versatile buffer to be used in biological system. Okay. So I don't think I need to explain this any further. We have discussed about pK multiple times now. So still, if anyone has any doubt, feel free to ask me, do not hesitate. I hope the concept of pK is clear now. Okay, so the correct answer is option B. Moving to the next question, calculate the mass of NaF that must be added to 300 ml of 0.25 molar HF solution to form a buffer solution having a pH of 3.5. We have the dissociation constant of HF given as 7.2 into 10 raised power minus 4, and the molecular weight of NaF given. So how do we solve this question? So we have here, what do we have here? We have a acid and a conjugate salt. So we have the acid HF and we have the salt NaF. We have to calculate the mass of NaF, that must be added to 300 ml of 0.25 molar HF solution to form a buffer solution having pH 3.5. So what equation are we going to use here? What equation are we going to use here? We are going to use here the Henderson-Hasselbalch equation. If you remember, in one of the question previously, we discussed the Henderson-Hasselbalch equation, that was pH is equal to pKa plus log salt concentration upon acid concentration, right? So we have the pH value here, we have the pKa value. How do we have the pKa value? We have the dissociation constant, that means we have the Ka value. From Ka we can calculate pKa, just take negative log of Ka, this will be equal to pKa. Okay, so you have the pKa value, you have the pH value, you have the acid concentration, you can find the salt concentration. Let's see how to solve this. So you have the pH value 3.5, pKa value is negative log of the Ka value, then salt concentration upon acid concentration is 0.25. Now from this you get the salt concentration as 0.56 molar. Now in the question, if you see, the question says 300 ml and they have asked the mass of NaF. So mass of NaF, so if I get concentration in molarity, how do we calculate the mass? I told you what is molarity. Molarity is equal to moles per liter, right? Molarity is equal to moles per liter. So here I got 0.56 molar. 0.56 molars means 0.56 moles per liter. Now 0.56 moles will correspond to, if you multiply it with its molecular weight, you will have the grams, and since it is in per liter, we have to multiply it with the volume that is required. So we said 300 ml, since molar capital M is given as moles per liter, we will have to convert the unit to liter. So 300 ml will be equal to 0.3 liters, right? So mass of NaF will come to 0.3 into 0.56 into 42. 42 is the molecular weight, and the answer will be 7.06. So if you solve this, you will get the answer 7.06. That is option C. Okay. Shaheen, you got option D. You might have made some calculation mistake, I assume, because both the answers are very close to each other. So I'm assuming there must be some multiplication error. So just look into it. I hope you will get this answer if you solve it using the equation. It's not a very complicated question, just that hello. 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 Yeah. Okay. Sorry, there was some error. I don't know. Sorry for that. Yeah. So where were we? So I think there should not be any issue in this question. I hope everyone is able to understand how to solve this question. See, if I would have asked you to just calculate the concentration of salt, it would be very easy. You can just put the values in the equation, in the Henderson-Hasselbalch equation, and get the value. But since in the question it is asked to calculate the mass of the salt, so from the concentration you have to also calculate the mass. So that part you have to understand how to solve these molarity related questions. Okay. So every time if you have a molarity, just think that molarity is moles per liter. So whatever the value, whatever the volume is given, you have to multiply with the volume to get the number of moles in that particular volume. Then from number of moles, if you want to get the value in grams, so just multiply it with the molecular weight and you will have, because molecular weight is grams per mole. So if you multiply moles with the molecular weight, you will get the value of like mass of the compound. Okay. So just if you just know the meaning of the unit itself, you can easily solve these kind of molarity based questions. Okay. Any doubt in this question? If there is no doubt, we will move to the next question. Okay. So the next question, question number 17. A 300 ml of 0.5 normal HCl solution is mixed with 700 ml of 0.1 normal NaOH solution. pH of the solution is approximately, so you have to calculate the pH of the solution when HCl is mixed with NaOH. So 300 ml of 0.5 normal HCl is mixed with 700 ml of 0.1 normal NaOH. So how to solve this question? Let's see. So we have 300 ml of 0.5 normal HCl. That means what would be the moles of HCl? If I multiply 300 by 0.5, so we will get the answer in millimoles, right? We'll get the answer in millimoles because I am taking the volume in milliliters. If I convert it in instead of writing 300, if I write 0.3 into 0.5, that will give me the answer in moles. Okay, so here what will be the answer? We will get 15 millimoles. Then similarly we can calculate the moles of NaOH solution. For NaOH we will have 700 into 0.1, so that will give me 70 millimoles. Okay, so once I have the sorry just a second. So 300 ml of 0.5 normal HCl solution basically means 300 into 0.5, sorry, not 15, 150 millimoles, and then 700 ml into 0.1 will be 70 millimoles. So now if I react HCl with NaOH, HCl will release one proton and NaOH will release one hydroxide ion, right? So HCl will be dissociating into H plus and Cl minus, NaOH will dissociate into Na plus plus OH minus. So 1 mole of HCl will be able to neutralize 1 mole of NaOH, right? So 70 moles of NaOH will neutralize 70 moles of HCl. So remaining 80 millimoles of HCl will be there in the solution. Do you understand? So here we have the base as the limiting reagent. So NaOH is limiting. So that means 70 millimoles of NaOH will react with 70 millimoles of HCl. That will mean that 80 millimoles of HCl will be remaining in the solution. So whatever the pH we will get, since NaOH will neutralize HCl, so we won't there won't be any change in pH. Whatever the pH will be there, the pH will be due to the excess HCl that is present. So 80 millimoles of HCl, that means 80 millimoles of H plus ions. Okay, and these 80 millimoles of H plus ion will basically result in the pH. So 80 millimoles of H plus ion, and we have to see the total volume since this is in moles, right? So the concentration for finding the pH, we have to have the concentration, that is the H plus ion concentration. The H plus ion concentration will be in molarity or normality, right? So 80 millimoles of HCl in 1 liter of solution, because 300 ml of HCl was mixed with 700 ml of NaOH. So total volume is 300 plus 700, that is 1000 ml. Okay, so in 1000 ml, that is 1 liter, you have 80 millimoles. That is, the molarity is 0.08 molar or 0.08 normal. Okay, so 0.08 molar will be the molarity, will be the concentration of H plus ions. If you take the negative log of 0.08, you will have the answer as 1.096. So option C is the correct answer. Do you understand this question? I hope it is clear now how to solve this question. Okay. Okay, so moving on to the next question, question number 18. Say 15 ml of 0.2 molar HCl is diluted to 1 liter. The pH of that solution will be approximately. Again, solve it similar to how we solved the previous question. So we have 15 ml of 0.2 molar HCl and we dilute it to 1 liter. So I hope you all are familiar with the equation M1V1 is equal to M2V2, where you have the molarity of the first solution, that is 0.2 molar, volume of the first solution, that is 15 ml, molarity of the second solution that we have to calculate in order to get the pH. So for calculating pH, we have to get the concentration of H plus ions. For getting the concentration of H plus ion, we have to get the concentration of HCl. Okay, so we have the molarity of HCl, we have the volume of HCl, initial molarity, initial volume, final volume, we have final molarity, we have to calculate. Final volume is 1 liter, that is 1000 ml. Since we have taken this in ml, just make sure the units are equal in both sides. Okay, so this is 15 into 0.2 divided by 1000. So M2 will be 15 into 0.2 by 1000. Okay, so just solve this. Once you solve this, you will get the concentration of H plus ions. Once you have the concentration of H plus ion, just take the negative log and you will get the pH, right? So let's see what's the answer. So we have 15 into 0.2 divided by 1000, that is 3 into 10 raised to power minus 3 molar. If we take the negative log, we will get the pH as 2.52. Okay. So option D, yes, option D is the correct answer. Very good. So 2.52. I hope this is clear to everyone. So these are all similar kind of question. I have added these questions to this session just to make sure that you practice well enough to calculate these molarity related questions. Since the topic is buffers and solutions, you will have many more questions like these. I have seen your assignments also have these kind of questions, so practicing these questions will actually help you get better. Okay, so I have added the detailed solution to each question in the slides. I will upload these slides also on Google Drive and you will get the link. So moving to the next question, again similar to the previous questions only. Calculate the approximate pH of a solution containing 10 raised to the power minus 2 molar H2SO4. Now we don't have HCl, we have H2SO4. So there is a catch here. What is the catch? H2SO4 basically dissociates into 2 protons. It has 2 protons, right? And it's a strong acid. So unlike phosphoric acid, it will dissociate all the protons in one go. So H2SO4 will dissociate into 2H plus SO4 2 minus. Okay. So now the concentration of H2SO4 is given as 10 raised to the power minus 2 molar. So if you see 1 mole of H2SO4 is giving 2 moles of H plus ion concentration. So whatever the concentration of H2SO4 will be, concentration of H plus ion will be double. Okay. For HCl, the concentration of HCl was equal to the concentration of H plus ion, right? Concentration of HCl was equal to concentration of H plus ion, but for H2SO4, the concentration of H2SO4 will be equal to 2 times the concentration of H plus ions. Okay. So now if you see here, the concentration of H2SO4 is 0.01 molar, right? 10 raised to power minus 2, that is 0.01 molar. So 2H plus ion, that means the H plus ion concentration will be 0.02 molar. Okay, just multiply it with 2, because 1 molar H2SO4 is giving 2 molars of H plus ion. Okay, so 0.01 molar of H2SO4 will dissociate into 0.02 molars of H plus ions. Okay, now you have the H plus ion concentration. Calculation of pH is very easy, just take the negative log and you will get the answer. This is also simple. The only difference in the previous questions and this question is to get the H plus ion concentration. Okay, in all the previous questions you are taking the H plus ion concentration equal to the acid concentration, but in this case, in case of H2SO4, the H plus ion concentration will not be equal to the acid's concentration. It will be higher than the acid concentration, because the acid is now having 2 protons. Okay, this is clear. It's not very complicated, I hope. I hope this is clear to everyone. This will move me to the last question of this session, question number 20. pH of a solution containing HCl is 3. Approximate concentration of HCl is. Now we have the pH given, we have to calculate the concentration of HCl. So it's again the same thing. So we have pH is equal to minus log of H plus. So if I have the pH, I can calculate the H plus ion concentration, right? So once I have the H plus ion concentration, I can have the HCl concentration, because HCl concentration will be equal to H plus ion concentration. Okay, but in this question we have the answers in grams per liter, so you have to pay attention on the unit. The unit is in grams per liter. The answer that we will get from here will be in molar. So we have to convert that molar into grams per liter. Grams per liter means molar is, I told you what molar is. Molar is moles per liter, right? So, and what is moles? Moles is basically, we have the given mass upon the molecular weight, right? So if I multiply the moles with the molecular weight, I'll get the grams per liter. So whatever the molarity will be, let's see. So here you can see you have the pH as negative log of H plus ion concentration, that is 3. So your H plus ion concentration is 10 raised to power minus 3, and HCl concentration will be also equal to 10 raised to power minus 3. Now amount of HCl in 1 liter will be 10 raised to power minus 3 molar into the molecular weight, that is 36.5 into 10 raised to power minus 3. That will give you the answer as 0.0365 grams per liter. Okay, this is clear to everyone. Option A is the correct answer. So how to convert from molarity to moles to weight? So the relation between the volume, mass, molecular weight and molarity and moles, all these terminologies, if that is clear to you, solving these questions will be very easy, like you can solve it in one click. Okay, so I know most people who are working in the labs already, people use all those molarity calculators that are available online, but for it would be very beneficial for you if you understand how those molarity calculators that are there online are also working. Okay, so if you understand the meaning of molarity, meaning of moles, meaning of molecular weight, mass, and the relation between them, it is very easy for you to solve these questions. Okay. So this brings me to the end of this session. I hope this is very clear to you. I have added many questions of pH and molarity related things, because this is something you should, like, this is something that will build the base of this topic. Like, if you are working in a biotech lab, or if you want to learn about biotechnology, these calculations are something that should be very clear to you in the beginning.
Ma'am, please give the next slide once.
Solution slide, right?
Yes, ma'am.
So anyone has any doubt in all the questions that we have discussed? We have discussed 20 questions today. So the topic was good lab practices and then buffers and solutions. I have focused more on the buffers and solutions part, because that is something, that concept has to be clear. So in good lab practices, you can go through the slides that are shared, and I will also share my PPT with you, so you will have these questions also, plus you have your assignments. So for good lab practices, I would suggest just go through his slides. That is more than, like, those slides have enough information for you to understand. And for molarity based questions, any assignments, any online content, if you are getting, I would suggest try practicing. If you see any where there are questions given, like how calculate the number of moles in this solution, calculate the grams in this solution, so try to solve those. Even if it is very easy for you, keep practicing those questions, because that is something that builds the base. Okay. So let me know if you have any doubt. We have 5 more minutes left till 9. So if you have any doubt, we can have a I can clear that doubt. Ma'am, can you please show the slide of the third question? Okay, just let me go to the third question. You want to see the solution slide of the third question, Shazneen? Okay, so if there are no more doubt, we can end this session. Thank you so much for attending this session. We will have these sessions on Tuesday. Like, I will take these sessions on Tuesday. Other TAs will have their other schedules, so you can join any of the session. Yes, Prajakta, you have a question?
Ma'am, for disinfection, what do we prefer? Ammonium salts or the hydrogen peroxide?
For disinfection,
Yes, ma'am.
I think hydrogen peroxide would be the better choice.
But ma'am, we can also use quaternary ammonium salts.
Quaternary ammonium salts, I am not very sure on that. I'll have to check. For disinfection, hydrogen peroxide is generally, like, what I know is generally used.
But in notes, it's provided that we have to use quaternary ammonium salts.
So maybe both of them could be useful. I'll just check and I can let you know if you want.
Okay, sure, ma'am.
Yeah.
Thank you.
In the notes, if it is mentioned quaternary ammonium salts, then probably that can
But
help you. Yeah.
I have heard that we prefer hydrogen peroxide more. So.
Yeah, even like I am telling you from my laboratory experience only that we do prefer hydrogen peroxide, but if it is mentioned, I haven't personally used quaternary ammonium salt, so I don't know. But probably if it is mentioned, maybe that is also a good choice of disinfectant.
Yes.
Yeah. Anything else?
Excuse me, ma'am.
Yes. Yeah, tell me.
Ma'am, if I join the other faculty class, so the discussion will be on the same or will be different in different sessions?
So the discussion will be more or less similar in all the sessions. It's just for your convenience. We have 3 different sessions in this course. So it is your choice. You can join any of the 3 sessions whenever you are free. And so basically all 3 of us are responsible to take the problem solving session for that particular week. So this was the session for the first week. So we will be clearing your doubts, whatever doubts you will have, we will be clearing your doubts. So the sessions will be similar, so you can join any of the 3 sessions. That is up to you, that is up to your convenience. If you are free in any of the 3 sessions, you can join. And all the recordings of all 3 sessions will be provided to you on the portal. There will be a link where you can find all the YouTube videos and all the lecture content. Like I'll share these PPT slides also with you, so don't worry about it, you will get all the materials that are required. And Prajakta, I was checking about quaternary ammonium salts, so they are also used as uh, disinfectant, so they can be used to kill the bacteria and viruses, so they are also a good choice of.
Okay.
Yeah,
Okay.
I just did not know that, that's why
Uh,
H2O2 is something that is very commonly used. So like uh, for general disinfection we use 70% IPS sprays, so these things are very commonly used. So
yes, ma'am. But if the option came, what we will choose, like hydrogen peroxide or ammonium salts?
uh, like if you are asked between 2 options, then I am not very sure. If your lecture notes mentions quaternary ammonium salt is better than H2O2, then go for
No,
that.
lecture didn't mention that, but it mentioned that we can use both.
So then we can use both. So if you have a question that which is better, I don't think we have a definite answer to that. I think both the options should be correct in that case. If you still have such question in an exam, I'm pretty sure that they will solve the conflict based on giving marks to both the options if they are 2 separate options. So like I don't think there's a better option or worst option.